right, well, welcome everyone. Thank you for joining us today. If you joined our last session, then thank you for joining us again. And in case this is your first time doing an activity with us, we're going to do a quick introduction explaining who we are and what activity we will be doing today. So hello, I'm Sonia. I am a second year biohealth sciences student and I have a minor in chemistry.
Hi everyone, I'm Grace. I'm a third year majoring in biochemistry and molecular biology with minors in chemistry and writing. We are both students from Oregon State University and are the community service officers for Sigma Delta Omega, an academic science sorority on campus.
So our philanthropy or charity is associated with K through 12 STEM education. So we were very excited when we got the opportunity to help out with the STEM webinar series. And hopefully we are able to help all of you who are interested in going into a science-related field learn more about chemistry and botany through a fun experiment we have planned. And if anyone has any questions, please feel free to leave it in the chat before we start.
All right, so as you already know, today we'll be discussing leaf chromatography. Along with this experiment, we will learn about the chemical processes and molecular reactions happening within the chromatography, highlighting topics like chromatography itself and chlorophyll. We have provided an optional fill in the blank worksheet, which I am going to put in the chat as of right now. And if you would like to follow along with that, you're more than welcome to, or you can also fill that out later. We just want you guys to have an opportunity to complete that in case you want to refer back to the information we covered in today's session.
So now we're going to go over the materials for today's experiment. So you will need glass jars or cups, a mortar and pestle, or you can use a spoon, isopropyl alcohol, white coffee filters or paper towels, leaves, you want them to be multiple colors, Scotch tape, and a baking dish.
All right, so does anyone have any questions before we begin?
All right, so we're going to start our experiment. And you may want to do this experiment in your kitchen since you'll need a stove and it might get a little messy. So gather all of your leaves and sort them into piles based on the colors. You should have 3 different colors of leaves and make sure that you have 7 to 8 different leaves for each color. The more the better. And if your leaves are wet from the rain outside, just dab them with a paper towel or a cloth towel to dry them off a bit. And all scientists make hypotheses or guesses about what they think will happen in their experiment. So let's all make predictions about what types of pigments we will see on our chromatography paper in each type of leaf. So remember to write your hypotheses down. Stop sharing.
So we have some lovely leaves. Beautiful. All right. So once you have done that, you've dried off your leaves with your paper towels, we can actually begin to grind up the leaves. You'll want to boil some water first, so you can fill a medium-sized pot with water and place it on your stove. You cover your pot with your lid and set it on the burner and turn that burner to high and bring that water to a boil. Make sure you have an adult helping you out with this step so you don't burn yourself. You can also use an electric kettle, which is much easier. You can just flip a switch and your water begins to boil.
Now we will start with our first color of leaves. So I'm going to use these brown leaves. And I'm going to start with, so first you're going to want to tear your leaves into small pieces and place the leaves into the mortar and crush the bits up to release the juices from the pestle. And if you do not have a mortar and pestle, you can use, put the leaves in a bowl and then just use a spoon to kind of crush them. I'll show you here.
All right. The leaves pieces don't have to be super, super tiny. Like if you just rip like pieces like that, maybe break that off like a little bit, that's perfectly fine. Yep, and you can use any type of leaf that you want from any tree, any color. Anything should work with this type of experiment.
It's a bit of an arm workout.
It really is.
So I think Emily asked, does it work better with crunchy leaves or soft leaves? And I think crunchy leaves work a little bit better, so that's why you want the leaves to be a bit dry. So I was able to dry mine off a bit with the paper towels, and I think it's crushing a little better now. Somebody asked, does it matter what color we start with? Nope, choose whichever color you like.
Yeah, and they don't need to be, you're not grinding it into a pulp or anything. This step is just so you can release the juices from the leaves. We're not here for hours. All right. I think mine are looking pretty good, Sonia. How are yours looking?
I think mine's looking pretty good too.
Okay.
It smells like fall.
It really does. This is fall right here. So I'm gonna show my mortar and pestle to the camera. I don't know if you can see that very well. It's a little dark, but you can see the leaves are ground up a little bit and they're starting to turn where the juices are being released. Perfect. All right. So once you're done crushing your leaves, you can place them into a glass, and we're going to repeat this process with your next color of leaves. So just like this.
I'm just using a glass that I found in my cabinet.
Perfect. All right, what color are you doing next, Sonia?
I do yellow.
Oh, me too.
I like your red leaves, Grace. I wasn't able to find any red leaves in my backyard.
Thank you. The street that I live on has a ton of trees, and all of them are like these red trees with these leaves. They just grabbed a giant handful of them this morning. Yeah, we have like giant piles of leaves on our street right now.
It's the season, I think.
It really is. Our whole backyard is just covered with leaves. I'm sure many of you. Yeah. All right. Yep. Grind it away again. Right, this is kind of an arm workout. Those of you doing it with a spoon and a bowl, it is a little bit more difficult, but I promise you it does work. Yes, today we're definitely going to be learning about why leaves change color in the fall and the different pigments within the leaves. Okay. I think mine are pretty good and crushed up now.
I think mine are too.
Yay.
Yes. These are my yellow leaves.
Ooh, oh, I forgot to do. There we go. Got ahead of myself.
Right, and I'm gonna put these in separate glass.
It would be an interesting experiment, seeing what happens if you put them together. Your results probably wouldn't be as good, but.
Yeah.
All right. Are we all set for the next step?
Yes, I think so.
Yay.
All right, step 4. So this is the stinky part of the experiment. So we're going to carefully pour the rubbing alcohol into each glass until the crushed leaf bits are covered or submerged.
So just press them down towards the bottom of the glass. So don't have to use as much. If you were using a spoon, you can use that spoon to push them down in there. All right. So just for you guys, this is a reference of how much rubbing alcohol I'm using in this glass. You can see that the leaf bits are completely submerged.
Yep, same here.
Yay. So now, what do you guys think the purpose of a rubbing alcohol is? So just like before, when you made your hypothesis about what pigments you'll see, it's time to come up with your own answer. So do you see like the alcohol doing anything to the leaves right now? You can go ahead and write that down. So the whole point of a rubbing alcohol is it acts as a solvent that helps to dissolve the leaves so that we can later extract that pigment from them. So moving on to the next step. You should have your baking dish with you. It doesn't have to be super big, and you can place your glasses carefully into your baking dish and make sure that they are equally spaced apart.
So next we're gonna take our boiling water and we're gonna put it around the cups in the baking dish. And maybe you can predict again what the boiling water does and what it does to the reaction. And then once you've made your prediction, we'll continue on. And by warming the rubbing alcohol, we speed up the process of drawing the pigments out of the leaves. So we can also cover the glasses with plastic wrap and wait overnight for the pigments to dissolve in the alcohol. I can definitely smell the alcohol now.
Oh yeah, it's pretty pungent. All right. Make sure you're careful with that step. You don't want to burn yourself. So you can actually set your glasses aside, and after about 30 minutes, we can see with the leaves, I believe pigments have colored the alcohol. I did this half an hour before our experiment, and this is what it should look like with the yellow leaves. You can see that the pigment, or not the pigment, the alcohol is dyed yellow right there.
I'll stop sharing real quick.
Oh, should I show it again? Is that better? Yep. The alcohol is pretty yellow now from extracting that pigment out from the leaves. So you will get the best results if you do this, but I was able to do this experiment successfully without soaking the leaves and rubbing alcohol for an extended period of time. So you can continue on with the next part of the experiment if you really want to. Okay.
Right, next step. All right, next we're gonna cut 1 by 5 inch strips from the white coffee filters, or you can use paper towels. I'm gonna use paper towels today. And you may adjust these measurements depending on how tall your glass is. I think I had to make mine a little longer because I have tall glasses. Um, but you will place one end of, into the solvent, so into the alcohol solution, and then drape the other over the edge of the cup. So I'm gonna assume this sat for 30 minutes.
All right. Perfect.
Yeah, we ended up starting this experiment in the afternoon and just left it overnight. But an hour or two sitting like this should be totally fine.
And then in the morning, you can pull up the filter strips to see what happened. The alcohol will carry the pigments from the leaves all the way up the paper and separate them into individual bands.
I'll share my screen here to show you the end result. Okay, so this is what your results should look like. And please don't be discouraged if you don't see your bands right away, or if it does not work for you. It does take a little while. So this is part of the scientific process, and the scientific process often requires you to adjust your methods, make corrections, or even apply those corrections to your next attempt, and it's all a part of the process.
Yes. And so I was able to do this experiment earlier, and then this were my results. You can see there's some darker bands at the very top and then some lighter ones down below. It's kind of hard to see, but there is some yellow near the very bottom. So I wasn't expecting that. And it's really cool to see the different bands coming out of the pigments. And that's another way that you can check to see if your hypothesis was right or not. All right.
So it looks like somebody asked if the bands aren't there, should we wait a little longer? Yes, so I think for us, since we had to leave it overnight, it took a little while for us, and I would leave it for maybe an hour minimum, just to see if the bands were starting to come up.
Yes, yeah, when I first started doing this experiment, even though I knew it was an hour, I was very impatient and wanted to see them right away and was worried it wasn't going to work. But I promise you, if you're patient with this process, you will see something eventually.
Okay, now let's go into the science. Okay, so now we're going to go over some of the fundamental principles of what makes this reaction possible. So the fundamental principles include chromatography and chlorophyll.
Great. So we've been throwing around this word chromatography a lot, and let's first break it apart. So the first half of the word chroma means color, and the last half, graphy, means writing or recording. So what this word actually means is that it's the method of separating the components of a mixture. So it's all about separating substances. And think about why this might be useful in some professions. We will discuss this in later slides. But during this procedure, the mixture will be separated into its individual components, allowing the individual parts to be identified. So in this case, we separated the pigments from the leaves and were able to identify those pigments on the chromatography paper.
Yeah, so the type of chromatography that we did is paper chromatography. And paper chromatography is a technique that involves placing a small dot or a line of a sample solution onto a strip of chromatography paper. And the paper is then placed into a jar, like in this picture, containing a shallow layer of solvent, and it's sealed. And the solvent is a substance that dissolves a solute, and a solute is a substance that can be dissolved. And as the solvent rises through the paper, it meets the sample mixture where it starts to travel up the paper with the solvent. Did we have another question? I think I missed it.
What are the other types of what are the other types of chromatography? I've only used paper chromatography in my science career, but I'm sure if you were to just do a quick search online, you'd be able to find it. Sorry, that's not a very good answer. But there are many other techniques for separating mixtures, such as I'm learning right now about separating different proteins, and you can use They're like separated based on like charge or their different environments that they're used to being in, such as pH. So this isn't the only method of separating different mixtures or molecules. There are hundreds of other ways you can do it.
And in our case, our sample solution and solvent were already mixed together, so they were able to travel up the chromatography paper together.
So there are three main factors that explain why different parts of the mixture separate as they do. So they are solubility, molecular weight, and polarity.
So first we will discuss solubility. So remember that the rubbing alcohol was the solvent for our experiment. So if the components of the mixtures are soluble in the solvent being used, the mixture will be carried up the paper strip as the solvent travels. If the material is soluble, these are tricky words, the mixture will dissolve as the solvent moves through. And if the material is a mixture of substances, some of these substances will likely be more or less soluble than others. So the more soluble substances will move faster and to a greater distance than those that are less soluble.
All right, so next we're going to discuss the molecular weight. Those substances of lighter molecular weight will move higher up the paper than those substances having a heavier molecular weight. So larger molecules have a harder time moving in the woven paper and get trapped in the paper first, whereas smaller molecules travel farther along the paper. So imagine like my hands are the fibers, and so it looks like this. And then if you have this really, like say these leaves are traveling like this, it's a really big molecule, it's gonna get trapped. But if you have a smaller leaf like this, which represents a smaller molecule, it's much easier for it to move through the fibers because it takes up less volume.
And lastly, the chromatography paper, as well as our coffee filters, is made up of cellulose, which is a polar substance. And the compounds within the mixture travel farther if they are non-polar. So more polar substances bond with the cellulose paper more quickly and therefore do not travel as far. So if you're confused about polarity, you can refer to our previous video, which can be found on the OSU Precollege Program's website. So on to chlorophyll. So the next principle we will address is chlorophyll. Have you ever thought about why leaves are green? Well, it is because of a specific pigment called chlorophyll. And leaves contain different pigments which give them their color. So chlorophyll gives leaves their green color and is essential for photosynthesis.
All right, so chloroplasts are known as organelles, which are specific structures in the cells and are only found in plant cells. This is one of their defining features as opposed to animal cells, and the chloroplasts contain chlorophyll. So chloroplasts are very complex organelles that utilize photosynthesis, the process that takes in carbon dioxide, what we breathe out, and water to make glucose, which is the food for the plant, and the oxygen gas, which is what we breathe.
However, chlorophyll is not the only pigment present in plant leaves. Xanthophylls are yellow pigments, and carotenoids give leaves an orange color. Chlorophyll usually hides the other pigments, except when autumn leaves come along and it begins to break down. And photosynthesis also uses these other pigments during the summer, but chlorophyll, a stronger pigment, overpowers them.
Right, so during the fall, when the length of the days become shorter and the temperatures become colder, this causes the leaves to stop making their food, and the chlorophyll begins to break down. So these pigments take more time to break down than the chlorophyll, referring to the orange and the yellow pigments, so you see them become visible in the fall leaves. So this is why leaves turn different colors in the fall.
Okay. So now we're going to talk about some notable women in science. So the first woman we're going to talk about is Catherine Isso, and she is a famous female botanist who worked at the University of California. She used botany, the study of plants, to study how various viruses affect plant development and the tissues within plants. And Isso was the sixth woman to ever be elected into the National Academy of Sciences, an organization that recognizes her outstanding work in science. and she later received the National Medal of Science from President George H. Bush in 1989 for her work in plant anatomy.
Yes, wow. And then the next woman we're going to discuss is Elizabeth Gant. She is a plant biologist and botanist and professor at the University of Maryland. Her work was primarily focused on photosynthesis, what we just discussed. Gant was the first woman to serve as the president of the American Society of Plant Biologists from 1988 to 1989. Like Catherine Issue, Gant was also elected into the National Academy of Sciences in 1996. Her pioneering role in plant biology paved the way for many other women to take leadership roles and positions in the American Society of Plant Biologists.
We are truly amazed by both of these women's achievements, and we are only touching on a small fraction of the amazing women in science.
Yes. So, if you enjoyed this experiment, you might be interested in careers that use biology and chemistry or botany. And you may want to be a research scientist when you grow up at research scientists design and conduct different experiments, usually in laboratory settings. There are many different types of research scientists. So do many do experiments with solubility and polarity, like we did, to develop new medications and identify unknown solutions.
And you may also be interested in becoming a botanist or a plant biologist. And botanists are scientists who study plants. And this includes topics like plant development, diseases, fungi, viruses, plant DNA, and so much more. Like in our experiment, some botanists study photosynthesis to understand and identify plant properties. Other botanists can work in agriculture or environmental science, where they study how plants can be used for food or how plants provide clean air to our environment.
All right. Well, we would like to thank you, all of us, for joining us today.
Yes, we hope you had a lot of fun and learned something new.
Yeah, if you have any more questions, use the Q&A function in the chat, and we'd be happy to answer any questions you have about this experiment or anything else that we do. Yeah, it should be safe for all ages. As long as if they're really young kids, you can have an adult present, and that should be perfectly fine.
Yeah. The only concern would be with using the boiling water and boiling your own water. So yeah, I think that would be a good area to have an adult with you in case you have a young student at home.
Yeah. If you're worried about the isopropyl alcohol, it's just rubbing alcohol, so that should be perfectly fine. Ooh, that's a good question.
The question was, what's your favorite plant?
Oh boy. I really like lupin. It's a really popular wildflower, purplish color. Yeah, I think that's my favorite plant.
I think mine are poinsettias. I think
Mm.
the holiday time is probably my favorite. So, and it seems very with the red and green.
Yeah. We have another question. Does it have to be boiling water, or is it warm or hot water good enough? It probably speeds up the reaction if it's boiling water, but if you feel more comfortable using hot water, that's perfectly okay. Yep. I also really do like succulents too. I have quite a few of them in my room currently.
Me too. A little too many.
I know. I was gonna say it's become quite an obsession of mine to just get a ton of succulents.
Yeah. I think they're very trendy now,
Mm.
succulents and houseplants.
Very, very true. 100%. Yeah. If you were interested in the types of chromatography that I was talking about earlier, they're called affinity and ion exchange chromatography.
Thank you, everyone. We hope you enjoyed.
Bye, everyone. Thank you so much.